Okay, welcome back. What I have here in front of us is long bone formation. And so we have this fetal bone development. And if I jump back to our guide, just kind of show you where we are. Um, let's see here. We're under the chapter 5 cartilage and bone. Let's skip over all the lovely cartilage. And then I kind of scroll down to bone development because I think this is some of the areas that we're struggling with a little bit. And I actually might go through all of these, but I started here with the first one for long bone development because we are looking, I believe, at primary ossification and not quite secondary. When you see this epiphyseal growth plate and this one, and probably even this one down here, we actually see both primary and secondary ossification for our endochondral ossification. Okay, so a lot of words. So let's come up here first to this one. And there's a guide over here to the side, again, showing us that we have our bone development and we get kind of this lovely view of, and it's very similar to the diagram I showed you in lecture 2, this view of the stages of long bone, right? And so we have this cartilaginous precursor, and then we want to end up with this adult bone last. But really, we're not going to really see that on a slide. We're not going to see an entire adult bone on the slide. And so what we do see is like a chip of the bone. So we can actually go in and chip this off. And that would be essentially your ground or your compact bone slide with all the osteons and the India ink that we saw before. And so where are we with this? So when we look at our slide, it's like, wow, there's a lot going on here. On your slide, it's probably more similar to this one down here. This is an entire limb, and so you can really see a lot going on with these long bones and even parts of the carpals and metacarpals that are up here. And so let's stick primarily just with the long bones, okay? And just and even if I use this, whoops, no, wrong direction. If I even scroll down here, we can see that this is a perfect bone we can look at. We actually can see the epiphyses, we can see the diaphysis, we actually can start seeing this band that is going to be the growth plate. And even if we use the ones up here, it's the same thing. We have kind of, again, that color and that textural shift that we always talk about. And you can see that there's this kind of darker purple, like at the epiphyses, and then this lighter that's happening at the shaft of the diaphysis of the bone. And so, at a low magnification, you got to get your bearings. You got to see kind of where am I? Where am I in comparison to this diagram over here? And you really are only seeing primary ossification. So these bones are showing you primary ossification, and then you would go to that epiphyseal growth plate slide to see both primary and secondary, which is a lot because your growth plate kind of takes over, and you might not have a lot of resting cartilage in that particular slide. And so you got stuff happening kind of like north and south of this epiphyseal growth plate. And what's also confusing on these limb slides, and this is essentially would be your fetal joint, right? So this is your fetal pig joint slide that you guys have, that it's sideways, right? And so we always want to look at, our brains go to this kind of north and south idea, like, what is at the top and the bottom of the slide? It always looks like a diagram. But in this case, you got kind of this, horizontal, an angled view that we're looking at. And so again, always having a diagram like what you see on the screen here, and also looking at your slide and having the ability to kind of go out to a bird's eye view and then back in. So go to 40, go to scanning, and then, click your way up into, low and high magnification. And if you get lost, go back out, to scanning, go back out to 4 power again and kind of figure out where you are. And so what else is on this slide? Well, we got, we have other tissue, we have potential muscle, you might run into skin. Look at, you can actually see skin right there. There's like stratified squamous right there. I don't know what's on your slide. Everybody has something that's a little bit different because they're all different sections, right? And so if we were to go ahead and kind of zoom in here, let's just use this guy. We can kind of pull this one up. We can already start to see that there is a difference. So we have all resting cartilage, and there might be a little bit of changeover happening here. This might just be the surface of the bone. We have mostly resting cartilage here too. And then we actually see that, there is some bony development, and we actually can see that those kind of look like spicules. They look like, trabeculae. That's our first stages of bone that we're making. So we have to make spongy bone first from this whole thing was cartilage, right? We have a whole pre-cartilage, cartilaginous precursor, right? So everything is cartilage, and then we have to kind of slowly change that over, and the epiphyseal growth plate is going to be kind of key in that changeover. And so all of this is resting cartilage, all of this is resting cartilage. We are going to start to see pockets of development happening right at the center of the epiphyses, and that's going to essentially start to turn over into to spongy bone, and that actually is what's happening here in the primary ossification area. We can go right to this lighter section here, this band of lighter purple, which is our growth plate, okay? And so when we go in real deep, right, and if you're in here, kind of like, let's see if we can go in a little bit deeper, you might kind of lose your bearings. And this is what I find kind of typical, that that confusion that happens. And it's like, which way is the growth plate going? And there's this kind of trickling effect of cells coming this way. There's all this big white space, and then you're heading up this way, and then things just kind of dissipate, and then you get really kind of consistent cellular material. And what is nice about this website is that they're also showing you where you are in the bone, and we don't have that ability in lab. You're in the slide, right? So you don't have something on the side that's kind of showing you where you are. And so if you're like, I'm getting lost, I don't know where I'm going, and you're kind of scanning around, that's what I'm saying. Like, go back out and get your bearings. Like, oh, okay, this is where I need to be, and then you can go back in, right? So kind of put the center of what you want to look at, right in the center of your field of view. And then we can, again, get our bearings. And so at this magnification, you can see lots of resting cartilage that is up here. It's all pretty uniform. And so we know that this is going to be resting. So this is the entire epiphyses up here. You might think, like, what is all this stuff to the side? Well, remember, there's lots of mesenchymal cells. There's lots of mesodermal nature cells that are giving rise to everything that is going to be from mesoderm, right? So you got lots of fibrous connective tissue. You have to make membranes. You got to make this joint. You got to make, all that connective tissue, and all of your spongy bone, and all the compact bone, and blood vessels, and all that good stuff is going to basically be happening here. And so you have lots of differentiation of these cells, but you have to remember, this is an embryonic slide, so we're heavy in development. And so if we were to kind of zoom in, this would be a great view of looking at like hyaline cartilage, right? And so that's something we could even do now. So all of this kind of watercolor wash in the background is going to be your hyaline cartilage. So there's lots of, collagen in there, of course. And we can have another whole video for this too. But just kind of as a quick reminder that this is all hyaline cartilage. We have lots of matrix that is here. Again, it has that kind of watercolor wash to it. We don't really see a strong presence of collagen, even though it's there, just with how we're staining. And all of these darker purple cells there are going to be our cartilaginous cells. Most of these at this stage are chondrocytes, and we know it's a site because we can see that lacunae around them. We have a space that is around the cell, and that is kind of our distinction marker that this is a site and not a blast. And so some of the cells you might be able to see a nuclei, some you might not. Maybe it's just kind of a uniform cell. So here's the cell, a membrane around the outside. It does have a nucleus, whether we see it or not, and cytoplasm filling the cell, the whole 9 yards. And then the space around would be essentially the lacunae. If I go back out in a little bit of a lower mag here, and I go to, let's see if we go to the surface, we might be able to see, oh, not really. Most of these are actually sites. So we can see kind of a distinction between the osteocytes and some of these cells here at the peripheral border that are similar to what we would think of as osteoblasts. And so even if I kind of come down here a little bit, let's kind of zoom into this area. Even at this particular location, again, lots of mesenchymal cells, but even at this point, you can see that most of these chondrocytes do have lacunae, right? And I'm going to go ahead and zoom out again, because what I want to do now is go down to the changeover. And so where we start to see that cartilage cells are going to change, and we have this growth plate that is pretty apparent. And so again, look for these color changes, textural changes, pattern changes. So even if this a slide, right, or photographs or black and white, you should be able to do pattern recognition that will kind of zone you in, right, pull you into what's happening. The color absolutely helps us, but we can definitely still see some of that shading difference, in like a black and white view. And so we can see up here all this lovely resting cartilage. And then we start to see that there's something happening, that there's all this white space that is starting to form. And what is happening is that the lacunae are becoming larger. And so that's telling us that there is a transitional section within this bone development. And so as these cells are starting to have their lacunae enlarged, they also start to organize into these parallel columns. So let's go kind of at the top area here. And you can see that, they're kind of scattered, but then all of a sudden they start to cluster, right? They're starting to kind of form these clusters or these islands. And then really even more so, you can start to see lines, right? Kind of strips of these cells. And so that's really what we refer to as those parallel columns of chondrocytes. And so when we start to see these columns, and yours might look a little bit different than this, and especially when you go to your epiphyseal growth plate, they look a little bit, I don't want to say they look different, but they might just be more uniform. They might be a little bit even more organized than what we see here. And so these columns are really the zone of proliferation. And so the cells are starting to they're preparing to basically kind of die off, honestly. And so these columns are the zone of proliferation. And then as we keep going down, right, we're kind of heading down that growth plate area. You can see that the lacunae are even becoming larger. And so we call this area the zone of hypertrophy. And so hypertrophy usually means something's getting bigger. And you also will notice that the cells are more difficult to see within the lacunae. And so every so often you'll see a cell here, but the lacunae are so big, they're kind of pushing the other cells out. It's just taking up more space. So you have essentially what looks like less matrix, this kind of light purple, like cartilaginous matrix that is here. So the cells themselves can start to disintegrate, they can get bigger themselves. There's just this kind of transition, right? So in that same section, you can see up here with the proliferative zone, you can see nuclei. But down here, you're like, where are the nuclei? Or you actually might see the cells getting kind of spidery looking as well. And that really is kind of the distinction between the proliferative zone and the hypertrophy zone. And a lot of times I get these questions of like, well, where exactly is it? And it's not, again, it's not a perfect science. You just kind of know this kind of zone of transition, like zones transition from one to another. And so to be safe, so if like you were to be asked, like, show me the proliferative zone, you would go somewhere up in here where all the cells are pretty consistent, lining up into these parallel columns. And then down here would be that kind of hypertrophy zone, right? The lacunae are really large, kind of pushing, they're kind of bubbling out. It always kind of reminds me of like blowing bubbles into like soapy water. They kind of like all kind of push out with each other. And then you kind of start getting down here. And this is really quite a line, right? This one is nice because you can see the zone of calcification. And so these cells really are starting to deteriorate. You get this kind of changeover. All of these bright red, or yours might be almost really a light bright pink or bright orangey cells, they almost glow a little bit. Those are going to be all of your red blood cells. So we see a lot of bone marrow within this region. Something else to note as we kind of continue down, because this is calcifying cartilage at this point, we're starting to have osteoblasts moving in. We're starting to calcify this tissue. It's basically called calcified cartilage. We're not fully making like this lamellar type of bone yet. Even spongy bone kind of has this rolling lamellar foundation to it, but we don't see it as true osteons like we do with compact. That's going to start to happen. So how do you identify? You're like down here like, well, what is calcified cartilage and where do we start to ossify? Again, there's a color change, right? So even if this was black and white, you would still see that there's a color shift. It's really helpful that it's in color, of course, but we can see that here is purple. And then what's happening down here? This is starting to become like more of a pink coloration, right? And so we have this kind of calcified cartilage. We're going to move essentially into ossification. And so as we travel down, again, we're kind of traveling down this way, we get to see that, wow, like we really are moving away from that cartilaginous matrix. We're really starting to see a shift over into this kind of bright pink color, and we can see potentially osteoblasts and osteocytes starting to form in here. We still have cellular death of cartilaginous cells. We have a lot of bone marrow, lots of blood vessels infiltrating this area. And then I get that question of like, well, what are these big pink strand things? Those are actually trabeculae. We actually start to form little bony spicules like the little ones, and then they start to fuse into bigger ones that essentially become the trabeculae. So you have trabecular bone or spongy bone forming here that will be remodeled into compact. All right, so we're not in that stage. We're not in that stage of development. So I get that question too. Well, like, where's the compact bone? Well, we're in the fetus, in fetal development. We don't see that yet. And so we want to see everything at the same time, but unfortunately we can't. And so that's why I say, okay, when you want to see compact bone, go over to the ground bone or the compact bone slide with kind of that India ink appearance, and then you get to see osteons and cross section. You have a slide that is longitudinal section, so you can see kind of both directions of the osteons that are there. And so would this be a great place to see bone marrow? Yes, right? These are bone marrow cells. This is a blood vessel kind of traveling through on the side. You get some red blood cells here. You get to see some bony cells that are trapped inside. And so we can kind of start differentiating because of the color. So if we're in kind of this pink area, we can start to say, okay, these are going to be osteocytes. The ones that are at the surface are going to be like osteoblasts. So because we're out of that cartilaginous area that was up above, right? If we go out to the surface, I'm just curious, if we go out to the surface, this is going to eventually become essentially the bony collar. This will be the actual compact bone density of the diaphysis. We're not there yet though, right? We're not there. We have lots of differentiation. This is going to become kind of like that surface. We can also see that will become an adult periosteum as well, right? And out to the side out here, we would start running into muscle tissue and all that kind of good stuff, right? So let's head back out in magnification real quick so we can kind of get an idea. Like we are really down here within the center of the diaphysis. And so, at the surface, look at how much more calcification to ossification is really happening. At the center, it's still like one step behind. And what you will see, and maybe on your slide you will have this, is that within this region here, it's going to really start to thicken up and become really dense, and it's going to continue all the way down that cylinder, right? That hollowed out cylinder that will become the diaphysis of your bone. And again, if I zoom out, we could do the exact same process on the other side, right? Because we have two epiphyses, we have two growth plates on these long bones. Again, it's important to remember that this is endochondral ossification, but not just anywhere. We have endochondral ossification in a lot of bones. It works just like this. Like you could see the same growth pattern when you were, if you were to look at your intervertebral disc slide, right? We went to intervertebral disc to look at fibrocartilage at the disc, but right on either side, flanking that disc, you also have your bone vertebrae. And so at that transition of cartilage to the bone vertebrae, you see this. You can actually see this happening there because that's endochondral ossification as well. We see this kind of two sides of the coin with a long bone because we can see primary ossification, and then when we pull the epiphyseal growth plate side, this epiphysis is starting to also go into the bone formation process, which is the secondary location, which is why we call it secondary ossification. There's also a difference because this bone, although it's spongy right now, look at all those beautiful trabeculae, that will be remodeled into osteons, right? It seems kind of counterproductive, like why wouldn't you just make osteons? But this is what we do. We make our spongy bone first, and then we remodel it into compact. Up here at the epiphysis, the secondary ossification location, this will just transition right into spongy bone, and it will remain spongy bone. What's also important to remember is that some of that cartilage is going to be retained, right, at the ends of the epiphyses. So you'll have kind of a layer of hyaline cartilage here. So all of this will essentially transition to spongy bone as an adult, but you will have a layer right along the surface here of hyaline cartilage, which then we kind of rename as articular cartilage, because that's where your bone would rub up with essentially another one, or that would be the purpose is we need a smooth surface, right? We need a smooth, somewhat not pliable, but we need a little bit of a spongier surface, and the hyaline cartilage will provide that. So we don't replace the entire surface with bone, right? So at the epiphyses ends, or at the articular surfaces of bones that make joints, we are going to contain or retain a little bit of that hyaline cartilage there. If I go back out a little bit and kind of scroll down, there's all this other tissue, right? And then you can actually see down here, like I mentioned before, this is like that stratified squamous. So you might have that on your slide. It's just upside down, right? So here's that keratinized layer, and this whole dark purple border is all of your epidermis. So that's all stratified squamous. And then it's running into the dermis, which will essentially be the skin, which is kind of cool. So you get to see that too. So let's go back out, whoop, wrong direction. Let's go back out real quick. Let's see if there's anything else that we need to take a look at. And I think that's pretty much it. What we also see on this slide, and also yours in the lab, that you can see there's a little bit of a space here. So that would be like a joint space that will develop, and there'll be, membranes developing. Your synovial membranes will be filling up with fluid. Again, you can see the space coming over to this bone. So you have one bone, you have another bone that is here. I don't think we get to see the entire one. No, probably not. We get to see a little bit of another one coming in here. But on your slide, you should have something actually look very similar to this. But your one bone should look very similar to this one. You should actually have two ends of that bone. For some of you, it's a very, very light staining slide. It doesn't have the depth and intensity that this slide does. It's very, very light. And so to those students, I tell you to work with your diaphragm lever and change how the light is coming through and refracting through that slide, because it actually helps to pop out some of the lacunae surfaces and like the border of the lacunae. So you might not be able to see a lot of this matrix. It might just have like a really, really faint pink appearance to it, which can be difficult for students to see. But what you can see is kind of the glowing outline of these lacunae, and that also does help you to identify the different zones within your epiphyseal growth plate area. All right, so I think I'm going to kind of pause it at that at this area. God, there's something I'm forgetting. Oh, the osteoclasts. That's the other thing. Sometimes we can see osteoclasts within our development slides like this, and they would be found right along the borders of the spicule to tuberculase that are forming when we're starting to remodel bone. And I'm trying to see if we have any on this particular slide. I think on the upper epiphyseal growth plate slide, there's absolutely going to be some of those osteoclasts that are there. Even that guy right there is starting to look like an osteoclast to me. They stain just a little bit differently, and maybe that one. But I know on the other, because a little bit further in development, we would see potentially more osteoclasts that are there. Okay, so that epiphyseal growth plate slide is where I'm going to tell you to also go. And developing cancellous bone or spongy bone, you can typically find some osteoclasts there too. Okay, so I'm going to wrap this one up and go ahead and look at the epiphyseal growth plate.